Knockout mice have specific genes inactivated or removed, while knock-ins have genes replaced or inserted. A way to develop such mice uses cultured pluripotent embryonic stem cells obtained from the inner cell mass of a blastocyst extracted from a female mouse that has mated. To distinguish offspring derived partially from the ES cells from those derived solely from the host embryo, the ES cells are obtained from a mouse having a coat color different from the mouse whose embryos will host the modified ES cells. The ES cells are genetically modified by inserting targeting vectors assembled by recombinant DNA techniques. To create a knockout, the targeting vector may consist of the neomycin phosphotransferase gene to disrupt the target gene and select cells with vectors. Two segments of DNA, called homology arms, flanking the DNA to be inserted so that a double reciprocal recombination with their matching sequences on a chromosome can occur and carry the DNA insert into the host cell's chromosome. A negative selection marker, like the HSV thymidine kinase, outside one of the homology arms to select against cells with the wrongly located insertions, and a restriction enzyme site between the vector backbone and the homology arm to linearize the recombinant vector. To create a knock-in, the vector is similar, but has the sequence to be inserted, cloned between the homologous sequence and the positive selection marker. The linearized targeting vector is delivered into the ES cells using electroporation, where a high-voltage electronic field creates openings in the phospholipid bilayer of the cell membranes. The targeting vectors, once in the cells, may undergo homologous recombination with the target loci in the mouse genome. There could also be random insertion. To select for cells with homologous recombination, the cells are cultured in medium containing G418 and gencyclovir. G418 kills the cells that fail to take up the vector, which have the gene that confers resistance. Gencyclovir kills cells where the vector was inserted randomly because the DNA inserted contains the TK gene. The surviving cells divide to produce clonal cells and can be sampled to confirm gene modification by running PCR and Southern blotting. Genetically modified ES cells are microinjected into the blastocyst from the mating between white mice. The modified embryos are then transferred into a female mouse induced to pseudopregnancy. Chimeric offspring that have some cells with dark fur derived from ES cells, which are heterozygous for the gene modification, are mated with a white mouse. Their fully dark offspring are heterozygous for the inserted DNA and are bred together to obtain homozygotes for the inserted DNA. Homozygous dominant mice can be differentiated from the heterozygous using PCR and DNA sequencing.